The single most infectious disease that infects 10 million people in the world every year and claims lives of 3 million people every year is tuberculosis. Unfortunately, 1/3 of this tubercular population lives in India, and therefore it is necessary to know the causes, the progress that is made in finding cures for this disease. And we are lucky to have Professor Colin Ratledge, who is an expert from University of Hull, with us today. We also have Dr. Bhaskar Saha from the Institute of Cell Science on the Pune University campus. And I would like to ask Professor Ratledge a question that in 1998, the sequence of the Mycobacterium tuberculosis has been discovered. What are the significances, what were the findings, and where can this lead us to in future?
One of the biggest problems that we've had is knowing what the organism that causes TB does when it's inside our bodies. The organism that causes TB is called Mycobacterium tuberculosis, and we have struggled for many years to understand how it is that it can lodge in our lungs, can multiply and divide, and now we are beginning to understand some of the factors which have to happen for that organism to succeed. And the important breakthrough came in 1998 when scientists throughout the world, but led from the Institute of Pasteur in Paris, were able to work out all the genes, all the biological information that was in the Mycobacterium tuberculosis, and have a complete sequence of the genome equivalent to what has recently been done for the human. So for the first time, we have a blueprint of what the organism is capable of doing.
As an immunologist, Bhaskar, what does this mean to you, and what would you like to know from Professor Ratledge?
Yeah, because genes encodes the proteins which may serve as very important immunogenic antigens. So it would be very nice to know how, knowing all those genes, having the complete blueprint of mycobacterial genome would help us designing vaccines and like getting the prophylactic vaccination schedules, etc.
Yes, one of the big challenges that we have if we're going to eradicate TB is how to develop successful vaccines. If we are going to develop drugs, we're treating cases of TB which have already occurred. But if we want to stamp out TB, then we're going to have to vaccinate everyone in the world, and that's the way that we can beat TB. The success of this strategy has already been seen with the eradication of smallpox. And smallpox has been eradicated by a worldwide vaccination program. So we know it can be done, and what we want for TB is a successful vaccine, and we don't have a successful one yet. We've got one which is okay, but it's not totally efficient. There are loopholes with it, and this is why TB is still on the increase.
In the past, children and we in our times were vaccinated with Bcg vaccine, and now we are told that it is no more effective. So what has gone wrong, or why it is not effective?
There's never been a successful vaccine. Bcg, even at its best, was only partially successful and gave protection to only about 50 or 60% of the population. So it's never been the perfect vaccine. If I can use the analogy with smallpox, the smallpox vaccine gave 100% protection. If you were vaccinated with that smallpox vaccine, then you would be guaranteed 100% resistance against smallpox. With Bcg vaccine, you can't make that guarantee, and therefore a significant number of people still get TB even though they've been vaccinated. It's only partially useful, but it's almost better than nothing. But really, if we could just get a vaccine that was as good as the smallpox one, we are on our way to beating TB permanently.
So now that we know the genes, I mean, exactly how is that going to help us designing a vaccine? Can you please tell us some of the ways?
Right. What happened with the BCG vaccine was that scientists in France about 70 years ago found a strain of the organism that came from cattle, Mycobacterium bovis. Originally it was called Mycobacterium tuberculosis, variety, bovis, meaning that it occurred in cows and gave cows TB. And they found a strain of that, quite by accident, which was safe to give to humans. And we now know that part of the genes, the genome, had been deleted accidentally. We don't know how, but it had happened. And now we had gone from this organism that would cause disease in cattle to having an organism that wouldn't cause any problems in humans, and it was a vaccine. And at the time it was used just as a first step. Unfortunately, there's never been a second step, and we're still waiting for the next vaccine to come over 60 years later. So now that we have the genome, we can look at what the genome is saying to the bacteria, and we can make some choices, and we can now delete some of these genes. And if we delete a particular gene, and we can delete many of them, some of those turn out to be very important for the organism to survive in the animal, inside the animal's lung. So we can now work out what are called virulence genes. If these genes are knocked out, and we can do this now in the laboratory, that converts the human Mycobacterium tuberculosis that would cause the disease into a harmless organism. And now this is the potential for building a new vaccine around not the cow bacillus, but against the human bacillus. So we can now turn the active Mycobacterium tuberculosis, which if I got, if you got, would cause TB in us, we can render it harmless. But it's still 99.999% complete. So it's got all the materials there which would then cause the immune response in the body to trigger and to become resistant to further infection. So the big hope is that we identify these genes which cause the organism to grow and multiply. These genes, if they're knocked out, stop the organism growing in the human body.
Is it possible to speculate when would this advancement in vaccine might take place?
It's taking place as we are speaking. Scientists around the world are now using this knowledge to develop vaccines, and I'm sure, again as we're speaking, some of these deletions, as they're called, from the human tubercle bacillus are being tried out in animal models. And it shouldn't be very long before we are reading somewhere in the press of people have been successful. There are groups around the world who are now trying this strategy, deleting what they believe to be the key virulence genes and working from there.
With this vaccination, we will be able to prevent the TB, onset of the TB. But you have to, would you be kind enough to tell what about those who have caught the TB? What options they have in terms of drugs to eradicate TB?
Right. Moving on from the vaccination program, which, let me just emphasize that if we do have the perfect vaccine, and we can have hopes that we can make the perfect vaccine, then we can eradicate TB. But it will take a long time. The people who are currently suffering from TB, and as you said in your introduction, there are many millions who are suffering from TB. We need new drugs to treat them. Now, unfortunately, the drugs which are used for the treatment of tuberculosis are very few, and the organism becomes resistant to them. so that it's no longer possible to kill them using these old drugs. And we are desperate to find new drugs that can be brought onto the market and kill the organisms.
Can I interrupt you briefly? The last new drug that was tried and which entered the clinical market was somewhere in the 60s, and there hasn't been anything new. Can you take a guess why this is so? I mean, apathy.
No, it's certainly not apathy, although there was a declining interest in beating tuberculosis amongst the major pharmaceutical companies. As far as the pharmaceutical companies were concerned, they felt that TB was eradicated in Western countries and there was little opportunity to make substantial amounts of money by developing new drugs. So the research almost died out in the major pharmaceutical companies across the world. But now we see the problem is returning, and it's returning not just in countries like India, it's occurring throughout the world. And it's a worldwide problem, and once more pharmaceutical companies are putting valuable resources into this and are developing new drug programs.
What are the new strategies that are coming in terms of finding drugs? Which fields, I mean, are there any major advances?
Oh yes, we go back to the genome, because the genome sequence tells us lots of important information. For example, we can identify which are these important virulence genes, and we know from the work which is currently going on that if we knock these out in the laboratory, we make the organism harmless. So it follows that we can make the vaccine, but also we can think about that gene which is making a virulence factor, which is a protein, which in all cases, or most cases, is an enzyme. And that does a chemical reaction in the tubercle bacillus cell. Can we find a drug that stops that key reaction working? So the genome tells us the target that the chemists should be looking at and saying, that's the enzyme that we've got to stop working. We know what the enzyme does, we know what it uses naturally, so can we find something that will stop it working and kill the tubercle bacillus? That's the hope.
But this has always been the practice, isn't it, Bhaskar?
Yeah, generating the inhibitors against the
But who is going to generate these inhibitors, and are there any advances on that front?
We are very fortunate that there's been a major revolution in synthetic organic chemistry, again over the past few years. Before, we've had teams of chemists working for many years to synthesize just a few thousand compounds. When I was a young man, I worked in the Medical Research Council in Ireland, where there was a serious TB problem. And I worked alongside a team of chemists who spent all their time synthesizing anti-TB compounds. And that team of 4 chemists, with technicians as well, synthesized about 2,500 compounds in 10 years. One of them turned out to be very effective and is used today in the treatment of leprosy. But that was a huge effort. Now the advance which has come in chemistry is called combinatorial chemistry. And the chemists can now make compounds by the dozen, by the score, by the 100, and by the 1,000. And now it's possible for a PhD student to make as many compounds in 6 months as the whole team of chemists that I used to work with made in 10 years. There's the advance. The other thing that's come along is the ability to test all these compounds. Again, when I was a young man working in Dublin, there was a team alongside the chemists testing these compounds. We now have what are called rapid throughput screening methods, and all these compounds can be tested within a few weeks. So it's wonderful advances. We are now at a position that we have never had before, with a combination of molecular biology, genetics and chemistry, the future for beating TB is looking extremely promising.
Well, with the revolutions in chemistry, genetics and molecular biology, there is a hope for not only eradicating tuberculosis, but treating it very effectively. On that hopeful note, and as Professor Ratledge had said, it's a cautiously optimistic view on the tuberculosis. We thank Professor Ratledge and Dr. Saha for being with us. Thank you.
Thank you.